Welcome, ladies and gentlemen. So today we're going to take a look at the meiosis drawing review worksheet. And we're going to trace the steps of meiosis using two sets of chromosomes. So we're going to keep track of who's homologous, who's not. We're going to take a look at crossing over between those homologous chromosomes and then follow those crossed over pairs throughout the rest of the diagrams, label some structures, indicate what actual processes are happening during each stage of meiosis, and then also indicate whether that stage is diploid or haploid. So if we take a look, prophase 1 is going to come first. We've gone through interphase, we've duplicated our chromosomes, and now we're taking a look at what's happening here in prophase 1. We'll start at what's happening at the beginning of prophase 1. So although crossing over is going to actually occur during this phase, we're going to write that in the notes, we aren't going to show the crossing over until we get to the next stage. So if I go ahead and come in here, I'm going to need to show two sets of homologous chromosomes. So I'm going to do that by just changing some colors. So let's just assume that that red is the one that mom would have given you, and then blue is homologous to that, and that would have come from dad. Now we're going to show two sets, so I'm going to do the same thing to another pair of chromosomes. And let's assume that the yellow is the one that mom would have given you, and the green one would have been the one that came from dad. Okay, now we're going to come in here and label some really important structures. So if I take a look here, all right, this is one of the two sets that we have of homologous chromosomes. So we're going to label this. And that being said, this set is also homologous to each other, but for the simplicity of just keeping our paper organized, we'll just label one set. Now they are also tetrads. I've got these two here plus these two here that are going to connect to each other and create basically a foursome or four-stranded chromatid set. So we can also call these a tetrad. Okay. And they're going to remain tetrads for the first couple of stages of meiosis before they end up splitting. Now, some other structures that are in here. All right, we've also got your centrioles going on here. These are where your spindle fibers are ultimately going to come from. We still, at least in the early stages of prophase, have our nucleus in place. All right, here in prophase 1, that will start breaking down. Just so we're clear, this is your cell membrane. Okay. And at this point, these guys still have their sister chromatids attached. And we're going to want to keep track of that as we go through this as well. Okay. So remember, sister chromatid is just one of these, and each of these duplicated ones has a sister chromatid attached to each other, and they are attached here at those center points. All right, and that is your centromere. Okay, so if we look at what's happening, well, first off, this is still a diploid stage or a 2N stage, because if you look, your homologous chromosomes, right, they're still together in a single cell. So that is what, by definition, a diploid cell would look like. Synapse is also occurring at this point, and that synapse is just a process of snapping together or connecting those homologous chromosomes. So they're going to form that tetrad, which is why we labeled that tetrad over here. So these guys are all directly connected to each other in this stage. And that's going to allow the crossing over to happen. We're not going to show it here, but we will see it in the next phase. I'll show you what those chromosomes crossed over will look like once it happens at the end of prophase 1. So remember, our drawing here is at the beginning of prophase 1. We'll show the end and what those crossed over chromosomes look like in the next slide. Your centrioles are going to appear, and they will start moving to the opposite ends of the poles or the ends of the cells as we move through prophase 1. Your spindle fibers will begin attaching to the centrioles and to the centromeres of those chromosomes as we move through this phase, and your nuclear membrane and your nucleolus will start to disappear and break down and will be completely gone by the time we get to the end of prophase 1. Okay, so metaphase comes next, and here we are going to show those crossed over chromosomes. So what's going to end up happening, and I'm only going to do this with one pair, again, just for simplicity's sake. So we're going to show one arm of that red chromosome being the parent, what it looked like from the original, and then the other half we're going to show a crossover. And again, this could happen multiple times to multiple places on the same location of the chromosomes, as well as between the different chromosomes we're dealing with, but I'm just going to show it once. Now, if it crossed over here, that means it also happened on the other side to the other homologous chromosome. So I've got a little bit of blue on that major red chromosome, and then I've also got a little bit of red on the blue one. Now again, I'm just going to show it on that set, so I'm not going to draw it on the other pair of chromosomes, but it could happen to these guys as well. All right, but again, just for the ease of making it through, I'm just going to show it once, but it would more than likely happen to both sets. Okay. All right, let's go ahead and label some structures again. So you still have this pair of homologous chromosomes. Okay. And they are still synapsed, or still connected together. So we are still going to call this a tetrad as well. All right, we've got our crossed over chromatid right there. Okay, where you've got the non-crossed over one on the other side. You still have your sister chromatids attached to those centromeres. You've got your centrioles hanging out here on either end, and those are being attached to these spindle fibers that are also being connected to those chromosomes. All right, so if I look, this is still diploid. You still have the homologous chromosomes together in the same cell. Your tetrads, or those homologous chromosome pairs, are now lining up in the middle of the cell. And we could add that label here too as well. If I kind of draw this imaginary equator or line down the center of the cell, all right, that is what we call your metaphase plate. Okay. Spindle fibers are going to finish their attachment from those centrioles to your centromeres on the tetrads. And don't forget, there is a checkpoint here for division, just like we talked about in mitosis. We want to ensure that those spindle fibers are attaching to all of those centromeres so that when we go to the next stage for anaphase, that those chromosomes split evenly so that the resulting cells at the end of this process get the correct number of chromosomes that they're supposed to have. We've also got independent assortment that's going to happen here. So when we look at these homologous chromosomes, I can align mom's information on the left and dad's information on the right, which is what I did here. But it's just as easily likely for this to line up like this, but to have your green and your yellow flipped. And either would be correct, either will happen when I go from one round of cell division to the next. It's possible for that to do that. And remember, that's one of the other reasons, in addition to that crossing over, that the cells that your body creates at the end of this process, whether they be eggs if you're female or sperm if you're male, that not every cell that your body creates is identical. And that's why if you have full-blood siblings, right, you and your siblings have the exact same set of parents, that you and your siblings don't look identical to each other. Because of the way that that independent assortment happens, you get a mixture of different traits mixed up on that genetic information when it gets passed, depending on which egg and which sperm get fertilized at each pregnancy. Okay, So let's take a look at anaphase. And here we're going to start to have those homologous chromosomes splitting from each other. So I'm going to have my red with my crossed over blue. Okay, and he's going to be over here. And then I've got that blue with a little bit of red. On this side, and then the rest of that chromosome is blue. Right? You can see that they're no longer attached to each other. And the same thing's going to happen down bottom where I have that yellow going one direction and the green starting to move the opposite direction. All right, so again, just to keep track of things, let's label some structures. Okay, so when I look at this, I still have my homologous chromosomes, so I'll still label those that way, but I'm not going to put the word tetrad here anymore. All right, and the reason we're not going to include the word tetrad anymore is because these guys have split. I no longer have 4 chromatids all connected to each other anymore. Instead, I've got these 2 sister chromatids connected, these 2 sister chromatids connected, but they're no longer in that 4-pair group. I still have my centrioles here. All right, those are still extending those spindle fibers. Your sister chromatids are still attached. Okay. And they're still attached at those centromeres. Okay, so when we take a look at this cell, because your homologous chromosomes are still together in that same cell, we're still talking a diploid cell here. Your tetrads, or those homologous chromosomes, are now splitting while the sister chromatids still remain attached. That's going to be important, and that's the reason we're going to have to go through a whole second round of division when we go through meiosis 2, because we want those to ultimately be separated into separate cells. And those sister chromatids still being attached are what's moving to the opposite ends of your cell, or what we call the poles of your cell, along those spindle fibers that are pulling them along. Okay, so telophase 1, there's no specific place on your worksheet to draw the cell, but I want to talk you through some important pieces, and you should add these notes to the worksheet around that telophase 1 label. So when we get to telophase 1, we're still talking diploid. Those homologous cell chromosomes are still in your cells, and you can see that if you take a look at the drawing that's up here. Your nuclear membrane, your nucleolus, those guys are going to reappear. They're going to come around the opposite homologous pairs and separate them into 2 separate nuclei. Your spindle fibers and your centrioles will disappear. And this will ultimately overlap with cytokinesis 1, which again also doesn't have a specific place to draw it. But cytokinesis 1, you're going to ultimately then split the cell down the center. That cleavage fur that you're seeing here is going to separate. You're going to have 2 distinct separate cells, and at that point, you no longer have homologous chromosomes in the same cell. And as a result, by the end of meiosis 1, specifically by the end of cytokinesis 1, those cells are now haploid, and they are no longer identical. Not only do they now contain half the genetic information from the original cell, so if we take a look at something like a human cell, we're talking 46 chromosomes to start with, you're now down to 23 chromosomes in each of these 2 cells. You've also got the addition of the crossing over and the independent assortment that have mixed up that genetic information between what your mom gave you and what your dad gave you before you go on to make the official egg or sperm by the time we get through the second round of division. That mixing up of genetic information has already happened at the end of this first round of cell division.